Hello students, this is the part 2 of the chapter Heredity and Variation. Now in the part 1 we learned about chromosomes. The chromosomes are mainly made up of DNA. So we will study what is DNA. Now DNA is deoxyribonucleic acid. In 1953, Watson and Crick they proposed the model of DNA molecule. Now DNA is present in the nucleus of the cell, and earlier it was also called as the nucleic acid as it was present in the nucleus. But later on Watson and Crick they gave a model of DNA, and we will see how this model looks like. So inside our body we have cells, and as our body is made up of cells, so in the cell we have the nucleus, and inside the nucleus we have chromosomes, and on the chromosomes we have DNA which has the gene, and the gene express our characters. Now this DNA model was discovered by two scientists, that is Watson and Crick, and according to their model, if you will see here, you can find that there are two parallel threads. Here we have two threads which are there, and these two parallel threads you can also call it as strands. So these are the two strands of nucleotides, and they are coiled around each other. Now as they are coiled around each other, this thing is called as a double helix structure. Now this structure can also be compared to a coil or a flexible ladder which we has two strands, and when you will just coil it, so you can find that it will look similar to this DNA structure. Now each of these strands, these two strands which are there, each of these strands are made up of many small molecules which are called as the nucleotides. And each of these nucleotides which are there, here we have the molecules of nitrogen base, we have the phosphoric acid which are joined by the sugar molecule. So in the DNA we have these three things. We have a nitrogenous base, we have phosphoric acid which is joined by the sugar molecule. Now how many nitrogenous bases can be there? Here how many nitrogenous bases can you see? So you can see four, that is cytosine, guanine, adenine and thymine. So there are four nitrogenous bases which are present, that is adenine, guanine, cytosine and thymine. Now the adenine and guanine here, that is A and G, these two are called as purines, whereas the cytosine and thymine, these two are called as the pyrimidines. So you have two things, that is purines and we have pyrimidines. Now these nucleotides which are there, these small nucleotides which are there, they are all the nucleotides, they are all like a chain. They are present in the DNA. It is arranged like a chain structure. So here you can see it is all in a form of chain. And the two strands which are there of the DNA, you can also compare it to the two rails of a ladder, and which are joined by the molecules of sugar and phosphoric acid. Now each rung of the ladder is a pair of nitrogenous base joined by hydrogen bonds. So here we have the nitrogenous base, so one here and one here, and these two, for example, this is adenine and this is thymine. So here adenine and thymine, these two are joined together here in with a bond that is called as the hydrogen bond. Now always you have to remember, adenine always pairs with thymine. So A will always pair with T, and the cytosine always pair with the guanine. So G will always pair with C. So you have to remember this, that in DNA the A always pairs with T and the G always pairs with C, that is adenine with thymine and guanine with cytosine. So this is the structure of the DNA, the strands which are present in our body, and this structure was given by Watson and Crick. Next we have the RNA, that is ribonucleic acid, and the RNA is an important nucleic acid of the cell. Now RNA, it is made up of ribose sugar, and it is made up of phosphate molecule and four nitrogenous bases, that is adenine, guanine, cytosine, and instead of thymine we have what? We have uracil. So this you have to remember, that in the RNA, we do not have thymine, we have uracil instead of thymine. So RNA is made up of ribose sugar, it is made up of phosphate molecule, and it is made up of 4 nitrogenous bases. But if you will compare it with DNA, so DNA is made up of phosphoric acid, which is joined by the molecule of sugar and the nitrogenous bases. Okay, so this thing you have to remember, that here we have a ribose sugar, we have a phosphate molecule, and we have 4 nitrogenous bases, that is adenine, guanine, cytosine, and we have uracil. Now the nucleotide, that is the smallest unit of the chain of RNA molecule, is formed by the combination of ribose sugar, phosphate molecule, and one nitrogenous base. Now large number of nucleotides are bonded together to form the macromolecules of RNA, and the main important functioning of RNA is the protein synthesis. RNA is very essential for the synthesizing proteins in our body. Now, according to the function, the RNA can be of 3 types. First is the mRNA, or we also call it as the messenger RNA. Now, as it is a messenger, it is like a postman. It carries information for protein synthesis from genes, that is the DNA segments in the cells, to the ribosomes, which is present in the cytoplasm, and where the proteins will be produced. So indirectly what it is doing, cell, in the cell, now there are nucleus also, we have different organelles also. So nucleus may say it is taking the information, and where it is giving that information to the ribosome. So nucleus में से जाके किसको information दे रहा है, ribosomes को, about what, about the protein synthesis. Next we have the rRNA, that is the ribosomal RNA. Now it is the component of the cellular organelle, so it is present in the cytoplasm, and the ribosomes, they perform the function of protein synthesis. So inside the ribosomes, the proteins are synthesized. Third, we have the tRNA, that is the transfer RNA. So as its name suggests, transfer, so it carries the amino acid up to the ribosome as per the message of mRNA. So mRNA क्या करता है, न्यूक्लियस में से मैसेज लेके जाके राइबोसोम को देता है, and then according to the message which is there on the ribosome, which was given by the mRNA, this tRNA, it brings the amino acid, and it gives to the ribosome. And all the amino acids, when they combine together, we get what? We get proteins. So this is how proteins are synthesized with different types of RNA. So we have 3 types of RNA. We have messenger RNA, which does what? It takes the message. We have rRNA, that is ribosomal RNA, जो क्या करता है? Which produces the protein. And then we have the transfer RNA, that is the tRNA, which transfers the amino acid according to the message of mRNA. Now students, we know that genetic material is transferred from our parents to us. But in what quantity these genetic materials are transferred? Now we get genetic materials from both our parents, that is from mother also and father also. But why only we get one character from the father or one character from the mother? Okay, so in what quantity these genetic materials can be transferred, and what will be the progeny, how the progeny will look like, what will the offspring will look like, that was given by the Mendel, which is called as the Mendel's principle of heredity. Now this was given by Gregor Johann Mendel. He was an Australian scientist. He had studied the inheritance of the characters of pea plant, and according to that pea plant, he gave his heredity, he gave his cross of how the characters can get transferred from one generation to the other generation, and what quantity these characters can be transferred, and how the progeny will look like. Let us first understand the monohybrid cross. Now, Mendel had studied about the 7 pairs of contrasting characters in pea plant. Now, here we are taking one pair of character, that is the parental height. So, if you will see about the plant height, so for first generation, that is parental generation, we can have phenotype as tall and dwarf. So the height of the plant can be tall, all the height of the plant can be dwarf. So what will be the genotype here? So it can be capital T, small t and small t. Now this capital T is the dominant character and this is called as the recessive character. So wherever you have, even if one single T is present, so the plant will be tall. And if you have one small t, then the plant won't be dwarf, the plant will be tall only. So now what will be the gametes that will be formed from here? So इसमें से एक t आया, इसमें से one small t आया. Then first generation that can be formed, that will be what? That will be capital T and small t. So first generation that we get, the first generation होगा if you make a cross between tall a pea plant and a dwarf pea plant, the plant that will be produced, that will be a tall plant only. Okay, now let us look at the parental generation 2. So for the second generation, now we have to make cross between this two tall plants. So here we are taking tall and tall, but now what are the genotypes? The genotypes are capital T and small t and capital T and small t. These are the two genotypes. So here we have to make a Punnett square. Here we will write the male gamete. This side we will write the male gamete. This side we will write the female gamete. So first gamete is what? Capital T and small t. So here we have capital T and we have small t. Then female gamete is what? Capital T, small t. So यहाँ पे capital T and यहाँ पे small t. Now make a cross. This and this. So the first thing that will be formed, that is the first cross that will be formed, that will be what? Capital T, capital T. So as both are the dominant character, the plant will be tall. Second will be what? Small t and capital T. So it will be what? Tall only because it is having one capital T. Next, this and this. Then we have capital T and small t. As one capital T is present, so it will be what? It will be tall only. Next is what? Capital T, small t and small t. So we have two small t's. That means the plant will be what? The dwarf. So that means how many type of parental or the generation that will be formed from these parental, that is P2. So we can either get 3, that is 3 is to 1 is the ratio. 3 chances are that the plant will be tall, 3% chances that the plant will be tall, and 1% chances that the plant will be dwarf if you make a cross between these, if you make a cross between this generation, that is the F1 generation. So this is how you make the cross with, and this is called as the monohybrid cross. So here we take only few characters, that is we have taken only one the one character here, that is the plant height. So we have taken only tall and we have taken only dwarf. So as we have taken only one character, it is called as a monohybrid cross. Next is the Mendel experiment on dihybrid cross. Now as it is dihybrid cross, so here we are taking two contrasting characters. So we have taken two types of characters. First is rounded yellow seeds and second is wrinkled green seeds. Now as it is rounded yellow seeds, the genotype is capital R, capital R, capital Y, capital Y. And as it is wrinkled green seeds, so the genotype is small r, small r, small y, and small y. So here what gametes, as all are same, so what gametes we can get? So one will be the rounded one, that is R, one will be the yellow gamete, that is Y. So we can get capital R and capital Y. Then here one is the wrinkled gamete, that is small r, and one is the green seed gamete, that is small y. So here we get small r and small y as the gamete. So now what will be the first generation that will be formed? So first generation, as these two gametes combine together, we can get first generation as capital R, small r, capital Y, small y. So that means this thing, that is as it is having capital R. So now which character will be the dominant one? That is the rounded one. So here it is rounded, and here as it is what? It is capital Y is the dominant character here. So it will be what? Yellow. So here the phenotype that will be formed of the first generation is what? Rounded yellow seeds. That means wrinkled green seeds the characters which were there of the wrinkled green seeds were the recessive character, and that is why they get suppressed. So the first generation that will be formed if we made a cross between these two things. So it will be what? It will be capital R, capital Y, small y, that is rounded yellow seeds. Now next we are taking P2 generation. So now what we will do, we will make a cross between the gametes which are formed here, the phenotypes which we got here. So from this we will make the gamete and then we will do a cross. So we will make a cross between these two, that is rounded yellow seeds. Now here we have the genotype as capital R, capital Y, small y. Capital R, capital Y and small y. So if you will make a cross, so here what gametes you can get? You can get capital R, capital Y, you can get capital R, small y, capital. Now all the capital R is done. Then next come to small r. So small r, capital Y, small r and small y. So these four gametes can be formed. You just have to make a cross between this only. ये इससे, फिर ये इससे, then ये वाला इससे, then ये वाला इससे. Then next we will come here, again same type of gametes can be formed here also. Capital R, capital Y, capital R, small y, small r, capital Y, small r and small y. So these are the gametes that will be formed for what? For the parental generation 2. Now let's put all the gametes, that is the male and the female gametes, in the Punnett square. So here we'll write the male gametes, here we will write the female gametes, whatever gametes we had found earlier, so we will write all the gametes here. Now when we start to do the Punnett square, you have to make a cross between these two. So for example, if I am making a cross, so first I will make a cross between this and this. So here it will be what? Capital R, capital Y, capital R, then we have what? Capital Y. So if we'll make a cross between this, what we will get? We will get two capital R's and we will get two capital Y. So that means what? This is what? A rounded yellow seed. So the plant which will be produced, that will be what? We will get rounded yellow seed from that plant. Next, we will make a cross between this one, this, and we will make a cross between this. So what we get? We get two capital R's, this one and this one. So we get two capital R's here, and we get what? One capital Y which is here and one small y which is here. So we get one capital Y and one small y. Now as this Y, it is a recessive character, so again it will be what? Rounded yellow seed only. Next we will make a cross between this and this one. So here what the phenotype we will get as? We will get one capital R, one small r and two capital Y. Okay, so now here as this R is again a recessive character and this capital R is a dominant character, so again it will be what? A rounded yellow seed only. Then next we will make a cross between this and this one. So then what we will get? We will get what? We will get capital R, small r and capital Y, small y. So that means again R here with this R which is present, it is what? A dominant character. This yellow is also a dominant character. So again we will get what? Rounded yellow seed. Now next we will make the cross between the next female gamete, and so this and the first male gamete. So here what phenotype we will get? We will get two R's, one capital Y and one small y. Again we have all the dominant characters, so it will be rounded yellow seed only. Now next we have to make a cross between this one and this one. So the characters which we get now, here you have to see, here you have what? You have two R's and you have two small y's. Now two small y means which character? That means two small y's are present. That means it is what? It is the green color which is there. So now what thing we will get? We will get rounded green seed. So same way you have to make the cross between this one and the next one, that is this third one, and this one and the last one. So we will get all the phenotypes in this manner. Then we will come to the third female gamete, you have to make a cross between again these two gametes. Then as we get the gametes, you have to write the phenotype, and then what you have to check is you have to check whether which dominant character is present and which recessive character is present. So here if we talk about, so here we have two small r's and we have two capital Y's. So two small r's, that means what? As there are two small r's, so that means the seed will be what? The plant which will be produced, it will be what? Wrinkled. And as we have two capital yellows, so that means it is what? Yellow. So we have wrinkled yellow seed. So here you will get wrinkled yellow seed. Same way here also you will get what? Wrinkled yellow seed. Why here wrinkled yellow seed? Because this Y is the dominant. If this won't be there, if it is small, then we will get what? We will get green seed. So in the same manner, we will do it for the fourth one as well. You have to compare, you have to make a cross between the male and the female gametes, and you will write all the phenotypes that can be formed. Last one that we will get what? We will get two small r's and two small y's. Now here no dominant characters are present. All the characters which are present, they are what? They are all the recessive character. So that means how the plant will be for the last one. So we will get what? We will get wrinkled green seed for the last one. So if you have to compare it, so here we get the ratio of 9, then we get the ratio of 3, 3 and 1. 9 is to 3 is to 3 is to 1. 9 we get rounded yellow seed. 3 we get the wrinkled yellow seed. Then 3 we will get round green seed, and 1 you will get as what? Wrinkled green seed. Now you can take other contrasting characters also and you can make a Punnett square and you can find out. So it can become easier for you to make the cross and find out how many number of phenotype and what can be the ratio of the phenotype. Usually the ratio of the dihybrid cross is always 9 is to 3 is to 3 is to 1.